Welcome back, everyone. So far in this series, we have explored the complete physics of infrared spectroscopy. We discussed molecular vibrations, selection rules, group frequencies, the Frank-Condon principle, FTIR instrumentations, interferometers, infrared sources, and detectors. But now we arrive at something every experimental scientist eventually learns. Even the most advanced instruments in the world cannot save a poorly prepared sample. Proper sample handling is absolutely critical in infrared spectroscopy. In fact, many bad spectra are caused by the instrument at all. They are caused by dirty sample holders, incorrect solvents, water contamination, poor grinding, or improper path length. Today we are going to learn how samples are actually prepared for IR spectroscopy. And then we will move into one of the most important practical skills in analytical chemistry, that is interpreting IR spectra. Infrared radiation interacts strongly with the covalent bonds. That sounds useful, and it is. But it also creates a challenge. Almost everything absorbs infrared radiation. Glass absorbs IR, water absorbs IR, many solvents absorb IR. Even fingerprint on your salt plates absorbs IR. So every materials involved in experiment must be selected carefully. A method depends on whether your sample is a gas, a liquid, or solid. Let us examine each one carefully. For gaseous samples, we use specialized gas cells. These are usually cylindrical evacuated containers with infrared transparent windows. Gases are diffuse. These molecules are far apart. That means probability of light interacting with the molecule is relatively low. So how do we increase absorption? We increase the path length. Many gas cells use multiple internal reflections. The IR beam bounces back and forth through the gas repeatedly before reaching the detector. This dramatically increases the interaction time between light and molecules. Remember the Beer-Lambert's law? Absorbance increases with the path length. Longer path lengths mean stronger absorption signal. Now let us move to the solutions. In solution spectroscopy, the solvent itself can absorb infrared radiations. This is a major issue. If the solvent absorbs strongly in the major region as your analyte, your spectrum becomes unreadable. Therefore, we choose solvents that are relatively transparent in important IR regions. Common IR solvents include carbon disulfide, carbon tetrachloride, and chloroform. These solvents contain relatively simple bonding patterns and exhibit fewer strong absorptions in critical regions. This minimizes interference. Now, here is one of the most important laboratory warnings in infrared spectroscopy. Never use water with standard alkali metal halide windows. Never. Most IR liquid cells use windows made from salts such as sodium chloride or potassium bromide. These salts are excellent for transmitting infrared radiation, but they are also water soluble. Water will literally dissolve your expensive optical window. And there is a second problem. Water itself produces an enormous broad area absorption, especially around 3400 centimeter inverse or 1600 centimeter inverse. These giant peaks can completely obscure your molecular information. To reduce solvent absorption, liquid sample cells are made extremely thin, typically path length are 0.01 to 1 millimeter. Very thin films reduce total solvent absorption while still allowing analyte detection. Now, suppose your sample itself is already a liquid. In that case, you may not need a solvent at all. You simply place a tiny drop of liquid between two salt plates. This is called as a neat sample. The liquid spreads into thin film and the IR passes directly through it. This is one of the fastest and simplest IR preparation methods. Now let us move to solids. Solid sample preparation is slightly more challenging. Historically, two classical methods dominate, the KBR palette method and the Mull method. The palette method, the solid sample is finely grounded into a powder. It is then mixed with potassium bromide powder. Potassium bromide is nearly transparent in mid-IR region, so it does not interfere significantly with the spectrum. The powder mixture is placed into a mechanical press. Very high pressure compresses it into a transparent disc. This disc is placed into spectrometer. Grinding is extremely important. Large particles scatter infrared radiation. This causes distorted baseline and noisy spectra. The sample must be uniformly fine. The second method is Mull method. The solid is finely grounded and mixed with a heavy mineral oil called nujol. This creates a thick paste. The paste is spreaded between salt plates, forming a thin film. However, nujol itself absorbs IR radiation. It produces characteristic peaks, especially in the carbon-hydrogen stretching region. So interpretation becomes slightly more complicated. Although not shown here, modern laboratories often use ATR spectroscopy, attenuated total reflectance. ATR dramatically simplifies sample preparation. You simply press the sample against a crystal surface. No palettes, no moles, no salt plates. This is one reason ATR FTR became extremely popular in modern labs. Now we understand the sample preparation. Let us move into spectral interpretation. Once you run the experiment, you obtain an infrared spectrum. The question becomes, how do we identify the molecules? One powerful approach is library matching. Suppose we obtain an unknown spectrum, we compare it against thousands of non-reference spectra stored digitally. Modern software performs peak picking, pattern recognition, and similarity analysis. If you are working with Python, you can automate this process easily using spectral libraries and correlation algorithms. The computer essentially performs a molecular fingerprint comparison, exactly like facial recognition or fingerprint scanning. Look at the comparison shown on this slide. The unknown spectrum is compared with the known benzene spectrum. When the peak position and intensities match, we confirm the identity. But what if molecule is completely unknown? What if no library exists? The infrared spectra are traditionally divided into two major regions, the group frequency region and the fingerprint region. The group frequency region spans approximately 4000 to 1300 centimeter inverse. This region contains relatively simple and characteristic stretching vibration. Functional groups absorbs here predictable weights. For example, oxygen and hydrogen stretches around 3200 to 3600 centimeters. Likewise, other groups also do. This region allows a rapid functional group identification. Even a quick glance can reveal whether alcohols, amines, carbonyls, or alkynes are present. Now let us move below 1300 centimeter inverse. This is called the fingerprint region. This region contains complex bending vibrations, skeletal vibrations, coupled motions, and whole molecule oscillations. The peaks overlap heavily. Unlike the group frequency region, individual peak assignments here are often extremely difficult. Even molecules with identical functional groups produce different fingerprint regions. For example, two alcohol isomers may both show oxygen hydrogen stretches, but their fingerprint regions will differ completely. That makes this region uniquely identifying, just like a human fingerprint. Successful IR interpretation combines both regions, group frequency region and functional group regions. The fingerprint region confirms molecular identity. When analyzing an unknown spectrum, step 1 is look for major functional group in the group frequency region, and step 2 is examining peak shapes. Broad peaks may indicate hydrogen bonding, sharp peaks often indicate isolated bonds. Step 3 is use fingerprint region for confirmation, and step 4 is compare against reference spectra wherever possible. Let me point out some several common mistakes students make. Mistake 1 is ignoring peak shape. Peak shape matters enormously. Broad oxygen hydrogen absorption look very different from sharp nitrogen hydrogen absorption. Second mistake is ignoring intensity. Strong peaks often correspond to highly polar bonds. Weak peaks may arise from symmetric vibration. Third mistake is forgetting solvent peaks. Always identify solvent absorption before interpreting the analyte. Mistake 4 is water contamination. Water contamination is one of the most common causes of confusing spectra, especially in KBR pellets. Infrared spectroscopy is used everywhere from pharmaceutical, pulmonary science, forensic chemistry, material science, environmental analysis, and biological research, and many more fields. A skilled spectroscopist can often identify the key molecular features within seconds simply by examining an IR spectrum. That ability comes from understanding both the physics and the chemistry behind the peaks. Let us summarize the today's lecture. Proper sample handling is essential for reliable IR spectra. Different IR preparation methods are used for gases, solutions, liquids, and solids. We discussed gas cells, solution cells, neat liquids, KBR pellets, and null techniques. We also learn how IR spectra is divided into group frequency region and fingerprint region. Together, these regions allow both functional group identification and molecular fingerprinting. In our next lecture, we will leave the world of infrared absorption entirely and enter another major branch of vibrational spectroscopy, that is Raman spectroscopy. There will there we will discover how molecular vibrations can scatter light instead of absorb it, and why Raman spectroscopy often reveals information that IR spectroscopy cannot. Thank you for watching. I will see you in the next lecture.